So hello everyone and welcome to our webinar, Golden Years of Semi-Automated Patch Clamp. So a warm welcome on behalf of the Nanion team. I am Marketing Associate Jaymar Portillo, and before we start, I would like to give you some general information about the webinar. By default, your video will be off and your audio will be muted. So this webinar would be recorded, as you probably have already seen. And by staying in this session, then you are consenting to being recorded. This webinar would be also later available on demand. For the Q&A sessions, they would be available after each speaker's presentation. And you can write your questions along the presentations. So we invite you to write them in the chat. But if you would like to formulate that yourself to the speaker, you can, after the presentation, raise the hand in the chat, and then the moderator will call your name and you can ask it yourself. And then at the end of the webinar, we will have a survey available for you, and we will appreciate very much your feedback. So a little information on the timeline of the webinar. We will have the first presentation by Dr. Patrick Mumm. Then second, we will then welcome Parsa Arman. And the third speaker of the webinar would be Elizabeth Kim. All right. So without further ado, we can then get started with the webinar. So it is my pleasure then to introduce you to my colleague, Dr. Patrick Mumm. Among his responsibilities here at Nanion, he is the product manager of the PortaPatch, of the product family of the PortaPatch. And he has been with Nanion for over 10 years. And it is our pleasure to welcome him today to tell us more about semi-automated patch clamp. So without further ado, the floor is yours, Patrick.
Thanks, Jaymar, for the kind introduction, and welcome everybody to this today's webinar. I just need to share my screen before I can get started. And yes, I just wanted to give you a kind introduction into the history of the PortaPatch and how it all started. So as you all know, the patch clamping is still the gold standard technique for studying the function of ion channels or electric currents transported by ion channels. With this technique, you place a tiny glass pipette on the cell membrane, you form a very tight seal and then gain electrical access by rupturing the membrane. The drawback of this technique is that it is a rather low throughput, it is very time consuming, and it requires significant training and technical capability, because you need to find your pipette tip under the microscope, you need to place it on the cell membrane, you need a vibration damp table, a rather big Faraday cage, and in general, quite a complex setup, which costs quite a bit of money. But in the 1990s, it became apparent that there was a need for a higher throughput in patch clamp technique, because several drugs were withdrawn from the market due to their side effects. And with this also mirrors a change in regulatory guidelines for drugs that came on the market. So there was a need to check those drugs on toxicogenic side effects or their potential on prolongation of QT intervals, for example, which resulted in the 2013 founding of the CIPA initiative, which will form a new paradigm of assessing pro-arrhythmic risks of new drugs. So in the late 90s, a bunch of companies were founded that were trying to put out the first automated patch clamp instrument on the market. So they all had similar approaches. Some were putting cells into a pipette, some were getting rid of the pipette, some were using loose seals instead of giga seals or population patch clamp approaches, but the most promising approach was getting rid of the pipette by using a planar glass substrate. The first thoughts about that were developed in the 90s, by 3 Ukrainian scientists. And to my shame, I didn't know that until about a couple of days ago, to be honest. But the first and the initial thought behind it by getting rid of the pipette is because no pipette, no hustle. You don't need the costly and heavy equipment like a vibration dam table, a huge Faraday cage. You do not need to pull your pipettes on a daily basis. You don't need a microforge and all that stuff. And the first successful recordings of cells on the planar glass chip were described by Niels Fertig, Robert Blick, and Jan Behrens in 2002. Niels Fertig, the good-looking man on the right side of the slide, and that led to the forming the foundation of the company where I work now almost a little bit over a decade. And when you do the math right, this was 21 years ago, and a year after the foundation of Nanyang, the world's first semi-automated patch clamp system saw the light of day, the Porta Patch, which is still until today the smallest patch clamp rig available on the market. It can record from one cell at a time, and it's very easy to use. It's very tiny, it has a very small footprint, but basically can do a lot of the things which you can do on a conventional rig, taking away the hustle of making pipettes and having that time-intensive training. So it's very easy to use. You can generate data basically on the first day you're working with it after having a short introduction. You can record voltage and current clamp on it like on a conventional rig. You can record whole cell and single channel ion channel activity. Due to the small volumes involved in the Porta Patch, there's a very versatile liquid handling. You have several add-ons, which I will mention in a little bit more detail later on. You can use it with the most amplifiers available on the market. You can use light as a stimulus for your cells, and we have consumables which are produced in-house, which can be customized. You also have the flexibility like you would have it when you make your own pipettes. Since the Porta Patch has been released, it has been adopted all over the world in over 20 countries, almost 100 universities and over 100 groups within academia and industry alike. And over 200 papers have so far been published with data of the Porta Patch, quite some of them in even high impact journals like Nature or Science. And the Porta Patch has shown his flexibility with a wide scope of different cells which were recorded on the Porta Patch, like primary cells or stem cell derived cells like cardiomyocytes or red blood cells. A lot of the cell lines that are available like HEK cells or CHO cells. You can record organelles on the Porta Patch like lysosomes or mitochondria, and even giant unilamellar vesicles. And with the variety of the cells you can record comes an even bigger variety of the targets you can record, like voltage-gated ion channels, like families of calcium and sodium channels, ligand-gated ion channels like ASICs or nicotinic acetylcholine receptors, and a lot more other channels and reconstituted proteins which you can put in your GUVs. The nice thing of the Porta Patch that you can tailor it to your needs by the nice add-ons that are available for the Porta Patch, like the external perfusion, where you can perfuse the external side of the cell you're recording with up to 8 different solutions to do precise dose response curve or activate ligand-gated ion channels. In this external perfusion, you can incorporate the temperature controls, so you can activate targets which are accessible for cold or heat activation, or if you want to record at physiological temperature. We have an internal perfusion where you can perfuse the cytosolic side of your cell with this tiny microcapillary which sits underneath the chip. We have a dedicated lid which fits on the Porta Patch, which can handle up to 3 different light sources when you want to stimulate channels like channel rhodopsines or work with caged compounds. And we even have a microscope slide which you can put under an upright microscope so you can see the cell you're recording in terms of checking your transfection rate or combining it with calcium imaging or things like this. And 20 years ago, the Porta Patch, as it was released, looked like this. And due to its proven success and robustness, we didn't change a lot in the last 20 years, so that the model now basically looks the same with a tiny facelift. But of course, the Porter patch is not there where the story of Nanyan stops. It was the starting or the start of Nanyan investigating and developing more instruments and broadening the portfolio, like fully automated multi-channel automated patch clamp devices like the patch liner or the synchro patch, or adapting new technologies like the surfer for transporter research, or the cardiac side to have a look at contractile cells like iPSC-derived cardiomyocytes. Or we have a dedicated instrument family dealing for bilayer recordings. And Munich nowadays has offices all around the globe. Like we have offices in America, in Germany, in Denmark, in China, and in Japan. And we want to be your one-stop shop for membrane science all over the globe. So if you're dealing with automated patch clamp cell analytics or membrane biophysics, I'm pretty sure we can be a good partner for your research. So that was it for a short intro from my side. And now I'm very curious to welcome our first speaker, Parsa Arman from the University of Texas. He has a background in biology and neuroscience and is currently a PhD student in the lab of Fernanda Leza. And we are very curious to hear something about IFG F14 peptide derivatives that are regulating sodium channels.
Thank you so much, Patrick. Can you hear me okay?
Yeah.
Okay. Should I just start sharing my screen? Let's see. So hello, everyone. My name is Parsa. I finished my master's here at UTMB Galveston in pharmacology and toxicology, and I would like to present one of my projects here in terms of studying these two ion channels. We utilize the Porter patch, and so I want to show you guys some of the things the Porter patch can do in terms of drug discovery and some of the work that I've done. So my title of my presentation is Intracellular Fibroblast Growth Factor 14 Peptide Differentially Regulates NAV 1.2 and NAV 1.6 function. So to give start of the presentation, what I wanted to do is I wanted to talk a little bit about how some of the current drugs and molecules that modify and modulate voltage-gated sodium channels work. Most of them bind to extracellular portions of the channel. Some of them bind to the pore, like TTX. Some of them bind to voltage sensors on the outside. And very few of them modulate the interior side of the channel. So there's a space to improve or develop novel therapeutics from that sense. And so here I have a picture of some of the different structures in terms of how drugs interact with various domains of the channel. Here are two really important antiarrhythmics that are known to be used in cardiology, antiarrhythmic 1C and 1A, I believe, and they're used widely. And so the main point of this slide was to kind of let you guys know that there's a lot of space in terms of real estate to be modulating these channels. And in addition, a lot of these peptides, and not peptides, a lot of these small molecules interact independently of auxiliary proteins. And so the auxiliary protein that we're going to be talking about is the intracellular fibroblast growth factors. These they lack the exportation signal on the protein, and so they're not thrown out into extracellular space, and so they remain intracellular and have a lot of interesting biological functions. And so from the inside, a lot of these FGFs can be phosphorylated via kinases, and they can be dephosphorylated via phosphatases. We do know that some of these FGFs can create homo and heterodimers with other IFGFs. Some of the other ones include 13 and 14, and I believe 12, 13, 14 are all intracellular. And some of them can even bind to microtubules. They can help stabilize the cell that way. And so what we're doing here is we're trying to understand the biology, the physiology of how these FGFs interact with the channel, characterize that, and and see which diseases may even have these IFGFs part of their pathophysiology, and how we can modify or modulate the interface between FGFs and sodium channels. So the FGF comes in, phosphorylated and unphosphorylated versions will bind to the channel, cause changes in the ionic current. And what we're trying to do is we're trying to throw something either in between the auxiliary protein in the channel and prevent them from binding, or we could create new pseudo IFGFs, essentially where a small molecule or peptide can bind to IFGF and cause a conformational change, and that whole complex can interact with the channel, giving you an interesting ionic signature. Or you could have a situation where the peptide simply interacts with the channel irregardless of the IFGF. In this case, that is one of the possibilities that we were trying to explore in this experiment. To give kind of like an overview of what sodium channels are, they're isoforms. So sodium channels come in 9 different flavors from 1.1 to 1.9. And interestingly, they are heterogeneously expressed throughout the human body, meaning that they're not all in the same spot and they're not all in the same cells. So they have cell specificity as well as location specificity. And these inherent built-in features of these channels can provide selectivity to certain areas of the body. And so we picked the 2 sodium channels in this study that we picked were NAV 1.2 and 1.6. And we had interest in the brain, the central nervous system. And so these are abundantly expressed. And one of the, interestingly, one of the example cells that we wanted to focus on were pyramidal cells. And they have a very interesting biological configuration of these 2 channels at the axon initial segment, which is the portion where action potentials are generated. And so biologically, these channels are put together in a very interesting layout where you have more 1.6. This isn't a mature cell because this would be different in developmental stages. So these channels change over time. But in a mature pyramidal cell, you have 1.6 more at the distal axon initial segment here, and you have more 1.2 relatively towards the proximal axon initial segment and more into the soma, into the dendrites. So from a perspective of understanding how action potentials propagate inside the cell, 1.6 is mainly responsible for propagating forward action potentials, while 1.2 is mainly responsible in coordination with other voltage gated sodium in coordination with other voltage gated channels like potassium channels, helps propagate back propagation into the dendrites. And so we wanted to see if our drugs that are modulating IFGF and sodium channels, if they have any ability to modulate the channel independently of IFGF. So, so we talked about the isoforms. We talked about how they're located inside the cell. And so getting even more granular, we'll talk about a little bit some of the mechanisms of voltage-gated sodium channels. So these channels have been studied for quite a long time, and they have various conformations, meaning they're not always conductive, and they have certain properties as how they conduct the flow of sodium channels through them. So at rest, at negative 80 millivolts, for example, the channel will be in a closed state. The voltage sensors within the domains 1 through 4 are situated in the plasma membrane and are not going to be sticking out because the voltage difference across the membrane isn't sufficient enough to drive the positive lysine charges that exist on the S4 segment outwards. As you slowly start depolarizing the cell, eventually you'll open up the channel, and activation is fast, and what we call this steady state activation. And as the channel is open quickly, as those S4 segments move out, an inactivation particle has access to its binding partners and immediately will inactivate the channel. But the channel can also exist, interestingly, in a closed inactivated state, meaning that its subthreshold depolarization is not sufficient enough to move the S4 segments out of the way. If, for example, maybe 2 S4 segments from 3 and 4, domains 3 and 4, move out, it isn't enough to open the channel, but it is enough to allow the binding partners of the inactivation particle to access, and therefore you can inactivate the channel without having opened it. And so there's a lot of interesting dynamics with this channel, and this is just speaking of the channel alone. We haven't talked about how IFGF can come in and modulate these. So the goal of this whole project was to identify compounds or peptides in this case that prevent the interaction between IFGF and sodium channel. So in A, we have a screening in which we've transfected hex cells with 2 different types of plasmids. One plasmid containing a CD4 segment, which essentially puts the so CD4 connected to the C-terminal domain, which is the portion of the sodium channel which we believe IFGFs are interacting with. We essentially put that into the cell, and now you have not the whole channel, but a portion of the channel sticking out into the cytosolic space. And then we have another plasmid containing the IFGF14 that has split luciferase attached to it. So one portion of the luciferase enzyme is on one protein and the other is on the other. And in the split luciferase compensation assay, if these 2 come together, you'll see luminescence. If they are disrupted, the luminescence will drop. And so we're testing these peptides that are derived from the interaction between IFGF and NAV1.6. So this valine we believe to be really important for that interaction. And so what we've done is we created a peptide scaffold called FLPK. And then out of that scaffold, we've created peptide derivatives, peptidomimetics, essentially, from FLPK. And we've created many of them, here's 20, and we've tested them in a split luciferase assay. And what we found is number 13, which is ZL017, has a really great C-log p-value, as well as a great ability to negatively control complex assembly. And so what we've done is we did a counter screen against the full luciferase enzyme. This would support the idea that these compounds are not interacting with, let's say, a catalytic site on the luciferase enzyme, and that in fact these peptides are preventing complex assembly. And we have cytotoxicity studies here, and we've done a dose response to calculate an IC50 value. Yeah. So what we've done is we've decided to do a selectivity study with this molecule to see if its mechanism of action is required, if its mechanism of action requires FGF to be involved, because that's where it was originally found. And we found that this peptidomimetic actually has intrinsically is able to bind to the alpha subunit alone and independently of IFGF, and it can have some effects. We're noticing that it has the ability to modulate peak current density. You see the IV curve here, in which we're giving, these are the 2 voltage protocols that we applied. The first voltage protocol gives us the IV curve and the peak current density and activation kinetics here. So this is steady state activation. And what we see is with 10 micromolar of this drug is we're seeing a hyperpolarizing shift in the V half of activation of this drug, meaning that this channel with this drug opens a little bit sooner in the action potential cycle, which is quite interesting. Continuing on, we also studied the inactivation kinetics, as mentioned earlier. So inactivation, this is steady state inactivation, in which the inactivation particle binds to its binding partners after the channel has been in the open state. And we're also seeing a hyperpolarizing shift as well. And so we have a very interesting phenomenon that we're seeing with this peptide. And in addition, we studied how this peptidomimetic interacts with 1.6. Again, we're seeing a very different type of layout here. We are seeing changes in peak current density, but we're seeing a depolarized shift in voltage of activation, steady state activation. And in terms of inactivation, we're seeing, again, the same sort of depolarizing shift. And so what we tried to do is we tried to rationalize these effects by looking at the mechanism of action of how currently sodium channels are thought to work, and kind of backtracking maybe where is this peptidomimetic binding and how is it causing its effects on the channel. And so we highlighted some spots that maybe we think are important to causing those effects, especially the S4 to S5 loops on the intracellular side, as those are connected to the voltage sensors. And if anything is pulling or pushing on these loops, they would essentially give you the signature of either hyperpolarizing or depolarizing activation. And we also have markations on the inactivation gate. And based on the literature, the first 100 amino acids that are coming out of the C-terminal domain are thought to be more important than the distal amino acids in inactivation. So for 1.2, we're seeing inactivation effects. For 1.6, we're not. So we're trying to rationalize that. And so we came up with a hypothesis as to where things are binding. And then we decided to test those in silico by doing by using AlphaFold. So currently there are no full structures, cryo-EM or crystal structures of the full sodium channel of 1.2, 1.6. So what we decided to do is we decided to use AlphaFold, which has never been done before, to render these channels and superimpose the structures onto segments that are crystallized. So there is cryo-EM of certain parts of the channel, but not the whole channel. So we decided to create a model here, and we decided to do docking studies, and we found that for 1.2, ZL has 3 hydrogen bonds to the inactivation gate, and has 1 hydrogen bond to the proximal C-tail. And so this kind of in relationship to 1.6, we're not seeing those strong hydrogen bonds with the inactivation gate, a very weak pi interaction here. And again, a hydrogen bond at the distal C-tail. So from these results, we're happy to see these results as they support our EFIS data and saying that the ligand has a stronger effect on inactivation gate, meaning that accounts for the effects of ZL on inactivation for 1.2 and not for 1.6. So to kind of wrap up my talk, so these channels are situated at the axon initial segment. And what this peptidomimetic is essentially doing is it's reducing the peak current density coming into the cell. And this, in hyperpolarizing 1.2 and depolarizing, shifting in a depolarizing manner of 1.6, this would essentially make these 1.2 channels open up sooner and make the 1.6 open up later. So this would have a very interesting effect in terms of how it would influence action potential kinetics. And we do have some in silico modeling of this, which I don't have shown here, but it will be in the paper because this is part of a manuscript that we are trying to prepare. And it'd be interesting to see not only the acute effects, but especially the chronic effects of this drug in terms of how the receptor profile of these channels would change over time. Maybe the cell will pull some 1.6 off the surface and replace them with another isoform, given the application of this drug over a long period of time. And so I just want to thank everyone in my lab that helped with these experiments. And I'm a new electrophysiologist. I've only been doing this for 2 years. And so the porta patch has been really, really beneficial in my learning, as I don't have the high level skill of an expert electrophysiologist. So the porta patch was very easy to use and very useful in conducting these studies. So I'd like to thank everyone for coming. And if there's any questions, I'd be happy to take them.
Thanks a lot, Parsa. Very interesting talk. And very good or nice for us to hear that you like the Porter patch so much because it's so easy to use and it contributed to your research. We actually have some questions here in the chat. Rod from our ES team was asking how did you add the compound? Do you use a perfusion system, or do you pre-incubate your cells before you do the recordings, or?
Pre-incubation, yes. We usually pre-incubate for around 30 to 45 minutes.
Another question we have in the chat: have you tried the effect of the protein on other channels located on the axon segment, like potassium channels?
No, which would be very interesting to do, because we do know that FGF13 does interact with some potassium channels. And so given that this peptide was derived from the interaction of the intracellular fibroblast growth factor and the sodium channel, there may be some overlap in the ability of this peptide to bind to other voltage-gated sodium channels. And in addition, this peptide may even have multiple binding sites on the isoforms that we were talking about, because when we were doing our AlphaFold modeling, we had multiple high probability sites that came up in the docking, and so what we did was we selected just the intracellular ones, but you know, who's to say that there aren't multiple binding sites for these drugs? And I think personally, that's good for drugs. If you try to be too selective with drugs, sometimes you lose efficacy that you're trying to have, especially when you get to patients and get to people and stuff like that outside of the lab.
Here's another question: is there a tissue specificity for different IFGFs?
Yes. I know that DRGs, not all DRGs so in DRGs there are 1.7 is kind of like the one that everyone's talking about, 1.82, but some DRGs express FGF13 without 1.7, but every time 1.7 is expressed in the DRG, I believe FGF is also expressed. And so yes, there are some cells that it can express FGF13, some that do not. But out of the exhaustive list of the entire body, I don't have them all memorized. But I know the nucleus accumbens, which is what we study in this lab, that is really a brain region that we're really interested in, does express FGF13. And in addition to that question, not all FGFs have the same effect in different cells. So you could be in one cell type, you could see maybe a reduction in peak current density, maybe a depolarizing shift. Another cell, you get a different effect. Maybe it reduces action potential propagation. In one cell, it increases it. Because these FGFs are not alone, there are other proteins that can bind to these channels. You kind of need to be thinking of FGF as kind of like one player in a massive complex. Some kinases like GSK3 beta can bind to these sodium channels and act as a scaffold protein. Another interesting protein that we've been studying in the lab that came out of one of my friends, he's studying creatine kinase B. So that's a very interesting protein, because we in the lab we kind of have this idea that metabolism is heavily linked to the expression of and modulation of these channels. So we found a creatine kinase B to also. So again, all these proteins, different disease states, different quantities come together and they can modulate these channels.
There comes an add-up to that question with the tissue specificity. What effect would the peptides have when administered systemically in vivo? Is there something you can say about that?
So I know a lot of people don't like peptides because they can degrade very easily. They're not very stable in the plasma. I don't have those studies, but I think we've done those studies. And again, this is not a true sense of the peptide, is a peptide derivative. So we took the peptide and we added different moieties to it. So, but you know, who knows? Maybe drug delivery in the future gets better. The drug delivery technology, and we're able to encapsulate these peptidomimetics in some sort of vesicle that can give us even more specificity, and we don't need the fact that the cells are hard to get to. We can, you know, maybe use an exosome. I don't know. We put certain proteins on the outside of the exosome. We can kind of send this, package it to a certain type of cell. But from the classical sense, yeah, I see what you're saying. Yeah, there's definitely an issue with plasma stability for sure.
And one last question before we're running out of time. Did you have a look at use-dependent effects of those peptides on the channels?
Like long-term inactivation? We did not run those studies, no. But I should have. I have the protocol, but we didn't show that information, no.
Okay, thanks. Then thanks again for your great talk, and we'll curious to hear another one from Elizabeth.
Thank you.
Like to welcome Dr. Elizabeth Kim from the Weill Cornell University. She is a postdoc in Krina Nimigan's lab and will tell us something about small binding sites of HCN channels.
Thank you for that introduction. I'm Elizabeth Kim. I'm a postdoctoral fellow in the Department of Anesthesiology at Weill Cornell. I just wanted to start off by thanking the organizers, especially Yamara and Irene, for the opportunity to give this talk. I'm also incredibly grateful to my advisor, Krina Nimigan, and the wonderful group of scientists at Weill Cornell, both past and present, that have and continue to shape the direction of this work. In addition, I would like to thank the structural biology centers where the cryo-EM data was collected, and also Roderick McKinnon for the constructs used in this study. Also listed here are the funding sources. And so today I wanted to talk a bit about my ongoing work centered on finding ways to regulate ion channel activity. The ability to activate or inhibit is advantageous for pharmacology and the ability to treat diseases. And so modulation of ion channel activity by small molecules has been largely categorized into 3 groups and have been shown to occur through multiple sites. And so here on the far left, many toxins and blockers produce their effect by blocking the pore. We can also have different ligands, such as cyclic nucleotides, that can activate channels. And there's also other small molecules, including anesthetics, that are thought to bind within the transmembrane domain, either within a single protomer or in between protomer subunits. And so again, the advantage of identifying allosteric sites over something more orthosteric that occludes the pore is in the ability to tune channel function that can either activate or inhibit. So for my channel of interest, HCN1 channels, they are tetramers shown here with each subunit shaded in a different color, and they are responsive to hyperpolarizing membrane potentials. And so because of their involvement in nociception and being primarily expressed in the central nervous system and not the heart, they are an attractive candidate for the development of a new class of analgesics. And so here I'm just showing a very small list of known HCN1 inhibitors and activators. These inhibitors include propofol, ketamine, and ivabradine. There are several more, but they are all pore blockers. Of the activators, the really only known activating ligand for this channel is cyclic AMP. However, alone, this cyclic nucleotide is unable to open HCN channels without the addition of some voltage across the membrane. Lamotrigine is a compound that was thought to enhance HCN currents, but was recently shown to have no effect. And so, as you can see, the majority of these compounds lack specificity and primarily target the pore. So while propofol is a well-known general anesthetic and its action is quite promiscuous across different types of proteins, it displays slight selectivity towards HCN1 over the other HCN isoforms. Therefore, we focused on propofol to understand its mechanism of action on HCN1, which is currently unknown. So what is known is that propofol slows activation of HCN1 channels. So here I'm showing some of the initial studies of propofol with HCN1. And this is actually 2 electrode voltage clamp data. And basically what they did was record currents at various voltages from Xenopus oocytes expressing HCN1 wild type channel in the presence and absence of propofol. And so in the black markers are the propofol data and the white markers are without propofol. And so what they observed here is that for the wild type channel, the V1/2 is approximately minus 60 millivolts. And this gets shifted to the left or to more hyperpolarizing voltages to around minus 90 millivolts. And in an attempt to narrow down the binding site, the channel was truncated to just the transmembrane domain. And parallel experiments showed that the same effect of an approximately 30 millivolts hyperpolarized shift happened in the presence of propofol. Therefore, it was concluded that this binding site is within the transmembrane domain. And so to determine its mode of action and to build upon this work, we wanted to determine the propofol binding site structurally by cryo-EM. And so I began this project by conducting a series of structural experiments for optimizing repeatable and observable association between HCN1 and propofol, for which I spent considerable effort. For these experiments, we had high concentrations of propofol that exceeded the IC50. And we also found that a lipid environment is necessary for stable incorporation of propofol with our channel. So we first set out to reconstitute HCN1 into lipid nanodiscs. And here on the left, I'm showing a representative chromatogram of this purification and reconstitution. And from this purified protein here on the top right are the typical micrographs of the vitrified protein on our cryo-EM grids. And from these images, we're able to isolate individual particles and categorize them into various views, leading to these 2D classes shown on the bottom right. And so from these 2D classes of particles, we are able to reconstruct the entire protein into 3 dimensions. And so here I'm showing you the final 3D map of our cryo-EM data of HCN1 with propofol. And this is at a resolution of 2.9 angstroms. And so in the end, we compared maps, EM maps between APO and propofol structures. And I was able to identify 2 densities that differed between the 2. And I have labeled them in this red-pink color. And they actually reside 5 to 20 angstroms from the inner membrane. So on the right here is the side view of our HCN1 channel. And on the left is a top-down view from the extracellular side, cross-sectioned here at this dashed line. And so one of the cool results from all these structural experiments we did was that when we increased the incubation time, we observed strengthening of the density for this one at site 1, suggested of greater occupancy. In addition, these densities range from the periphery of the channel, with site number 2 being on the outermost, and gradually move inwards toward the core, with site number 1 being closest to the pore axis. And so because site 1 has stronger density with increased incubation time and is closest to the region most important for channel gating, I will focus on this site for the remainder of the talk. And so there were 2 interesting observations with our propofol HCN1 structure. What I have here on the far left is an overlay between the APO and propofol modeled structures with a propofol structure in blue. The overall RMSD is 0.9 between the 2, which means they're very close in structure. But in the center and right panels, I'm showing the pore diagrams for the propofol structure, showing that 1, it stabilizes a closed state, as we expect since this is an inhibitor for the channel. However, this closed state appears to be a novel conformer with slight differences in the selectivity filter, and the lower gate is tighter with a more closed, being more closed at the glutamine 398. The second interesting thing is specifically in regard to site 1, and this site actually exists between 2 protomer subunits, shaded here in yellow and blue. And this pocket is where our putative propofol density exists, and this appears to be a state-dependent pocket. So I've outlined this pocket in the yellow dashed line here in each panel. And so on the far left is our HCN1 propofol structure with our propofol molecule in red modeled in. And the second panel shows that this pocket actually exists in the closed HCN1 structure published by the McKinnon group. However, this pocket becomes inaccessible or reduced in size in the structures in panel 3 and 4, where we have a cross-linked HCN1 hyperpolarized conformation or the open HCN4 structure. So the current model based on our structural data suggests that Propofol occupies a state-dependent pocket to sterically inhibit rotation of key helices, including the S6 helix for channel opening. And this is what I'm trying to depict here in the schematic of our cross-section top view. In the overlay, there's a steric clash between the closed and open states occupied by our Propofol molecule at site 1. And so next, in support of this model, we performed both 2-electrode voltage clamp on Xenopus oocytes or whole cell patch clamp using the porta patch of HCN1 expressed in hex cells to functionally validate our binding sites that we identified. And so here I'm showing data of hex cells expressing HCN1 wild type and the family of currents in response to different voltage steps. And as I mentioned previously, to look at how drugs modulate the voltage response of HCN channels, we care about the changes in V1/2. And so to measure this, we looked at the Boltzmann fits shown here on the right. And we do these experiments with and without incubation of 30 micromolar propofol. And so here I'm showing you an overlay of these Boltzmann curves for our wild type channel in the absence and presence of propofol labeled PFL. And as published previously, we confirmed that we see a hyperpolarizing or leftward shift of about 30 millivolts in the V1/2 with incubation of propofol with our cells. And so we did these parallel experiments with a few different mutations in our binding site 1. And this is a subset of them as shown in the plots in the different shades of blue labeled as mutation A and mutation C. And I hope you can appreciate that these two particular mutations reduce the delta V1/2 between the APO and propofol conditions. Therefore, we were able to knock down the effectiveness of propofol inhibition for these particular mutants. So here on the left panel is just a composite of the different mutations I tried. The size of these boxes represent the amplitude or magnitude of the delta V1/2 between the currents measured with and without incubation with propofol. And at least for the mutations labeled A through D, in general, we observed a reduction in the delta V1/2, and using some in silico structural mutagenesis prediction for these mutations, we actually found that these 4 mutants reduce the size of that binding pocket, and this is likely the cause of the reduced magnitude of inhibition of propofol for these mutants. And this is quantitated here on the graph in the right. And so the next question we asked was, how can HC1 be selective for propofol over the other isoforms? For this, we took a multiple sequence alignment to look at the binding sites we identified structurally across all 4 isoforms. The 2 sites we identified here are labeled PFL1 and 2, and I've highlighted the residues lining these binding pockets in these red boxes here. And from this alignment, you can see that they are very conserved. So the question becomes, how do we actually gain selectivity, or how is it achieved? There are 2 primary ways in which we can gain selectivity. The first is in the binding site itself, which does not appear to be the case here due to the conservation. The second is the differential allosteric propagation of the actual binding event. So to look more into this question, we took another approach to identify allosterically coupled residues by coevolution. This idea is founded on the principle that residues that display coevolution or changes in tandem with one another do so because of a functional codependency. So in the row labeled SCA in green, I've highlighted a subset of residues that actually show strong coevolution to positions within these cryo-YEM binding sites. Of these positions in the green boxes, I'm highlighting those that do not show absolute identity to the HCN1 residue, and these are positions of interest that I will look into the future experiments, in addition to further functional characterization of that second propofol binding site. So in conclusion, we have identified 2 putative propofol binding sites. They indeed are within the transmembrane domain, but we have now localized at least one of them to a pocket between the S5 and S6 helices. Our current model generated from this data suggests that isoform specificity is not from the residues that actually comprise the binding site, but from residues that are energetically coupled outside of that. So thank you for your attention, and I would be more than happy to take any questions.
Thank you very much for your interesting talk. We have a question in the chat. What's the difference between the HCN currents with and without N-terminus?
So the N-terminus primarily it's been actually shown that HCN channels don't really need the N-terminus that much. There's a HCN binding domain that is required for its proper trafficking to the membrane, but you can chop it off pretty well and still retain currents.
Here's a question from Juha. Have you tested any other compounds like ivabradine?
I have not. Ivabradine is quite a sticky molecule. The closest I've come to a pore blocker is just using cesium ions, cesium chloride.
Can you say anything about the propofol effect of CAMP affinity on HCN?
I have not done those experiments, but that is something I'm interested in looking at. I think there's a lot of crosstalk between cyclic AMP and the voltage gating.
Yeah, I guess then the question following this one is you didn't do the experiment. So does CAMP concentration change the propofol effect? But I guess that's nothing you have done so far.
I have not done that so far.
Another question we have here, since propofol is a general anesthetic, do you propose that it mediates its anesthetic effect via HCN1?
No, so the primary target for propofol for its anesthetic effect is the GABA receptor, and that's quite well known. So yes, this will never be probably used as a true treatment for anything, but it kind of gives us ground rules on how to design things that target HCN1 more specifically.
Did you have any look on the propofol effect on other HCN channels like HCN2, or did you?
I personally did not, but there's a lot of literature looking at the other HCN channels. Yeah.
Then thank you very much. I think that were all the questions so far. Thanks to you, Elizabeth and Parsa, for the nice talks we had. I, in the end, would like to still have one information which I wanted to share with you, since some of our listeners maybe are now intrigued to get their hands on a porta patch. Due to the 20th birthday of the porta patch, we are offering the porta patch with a 20% discount till end of the year. So if you are interested in getting a porta patch on your own to accelerate your research, then please be in touch with one of our team and we can discuss a demo, or if you need a quote, just let us know.
Yeah, and also before we go, I just wanted to show you also just one slide. So first of all, thank you to Elizabeth and Parsa for showing the interesting talks and results from their projects. So we are very looking forward for the publications to come out and read them. And yeah, so before we go, I just wanted to let you all know that we have in our website, in the news section part, you can find the upcoming events And basically, I wanted just to highlight some of the upcoming ones. So we have another webinar on SSM electrophysiology on the 20th of July, and also to bring some attention to our Nanion user meeting. This is happening in October, the 24th and 25th of October. So this would be a hybrid event, and you would be able also to join remotely. Yeah, and last but not least, we have yet another webinar about high throughput electrophysiology that would also happen in July, the 13th of July. So just if you want to hear about our news, so you can always go to the website, or you can follow us on the social media channels so that you stay up to date. And even better, you can also subscribe to our monthly newsletter so that you get all the information right on time. So thank you all again. And I just wish you a nice day, evening, depending on when you are. Thank you and take care.
Bye-bye.